Role of mitochondria in the formation of ATP. Mitochondria are often described as miniature power plants. Like power plants, mitochondria extract energy from organic materials and store it in the form of electrical energy. More specifically, the energy extracted from substances is utilized to generate an ionic gradient across the inner mitochondrial membrane. Mitochondria utilize an ionic gradient across the inner membrane to drive numerous energy requiring activities, most notably the synthesis of ATP. When ATP formation is driven by energy that is released from electrons removed during substrate oxidation, the process is called oxidative phosphorylation. Oxidative phosphorylation can be contrasted to substrate level phosphorylation, in which ATP is formed directly by transfer of a phosphate group from a substrate molecule to ADP. Oxidation reduction potentials. The early work of Warburg and Kalin using spectrophotometric techniques led to the concept that cell oxidations are brought about by electron carriers arranged in a chain of oxidation reduction or redox potential. Since the movement of electrons generates a separation of charge, the affinity of substances for electrons can be measured by instruments that detect voltage. Protein import and mitochondria assembly. In contrast to the RNA components of the mitochondrial translation apparatus, most mitochondrial genomes do not encode the proteins required for DNA replication, transcription or translation. Instead, the genes that encode proteins required for the replication and expression of mitochondrial DNA are contained in the nucleus. In addition, the nucleus contains the genes that encode most of the mitochondrial proteins required for oxidative phosphorylation and all of the enzymes involved in mitochondrial metabolism. The proteins encoded by these genes are synthesized on free cytosolic ribosomes and imported into mitochondria as complete polypeptide chains. Because of the double membrane structure of mitochondria, the import of proteins is considerably more complicated than the transfer of a polypeptide across a single phospholipid bilayer. Proteins targeted to the matrix have to cross both the inner and outer mitochondrial membranes, while other proteins need to be sorted to distinct compartments within the organelle. The import of proteins to the matrix is the best understood aspect of mitochondrial protein sorting. Most proteins are targeted to mitochondria by amino terminal sequences of 20 to 35 amino acids that are removed by proteolytic cleavage following their import into the organelle. The pre-sequences of mitochondrial proteins, first characterized by Gottfried Schatz, contain multiple positively charged amino acid residues, usually in an amphipathic alpha helix. The first step in protein import is the binding of these pre-sequences to receptors on the surface of mitochondria. The polypeptide chains are then inserted into a protein complex that directs translocation across the outer membrane. The proteins are then transferred to a second protein complex in the inner membrane. Continuing protein translocation requires the electrochemical potential established across the inner mitochondrial membrane during electron transport. The transfer of high energy electrons from NADH and FADH2 to molecular oxygen is coupled to the transfer of protons from the mitochondrial matrix to the intermembrane space. Since protons are charged particles, this transfer establishes an electric potential across the inner membrane, with the matrix being negative. During protein import, this electric potential drives translocation of the positively charged pre-sequence. Proteins are targeted to the Tom complex in the mitochondrial outer membrane by amino terminal pre-sequences containing positively charged amino acids. The pre-sequence first binds to Tom 20 and Tom 22 and is then transferred to Tom 5 and the import pore Tom 40. Following passage through the outer membrane, the pre-sequence binds the intermembrane domain of TOM22 and is passed to the TIM23 complex in the inner membrane. In the matrix, an HSP70 chaperone associated with TIM44 acts as a ratchet using ATP hydrolysis to translocate the protein across the inner membrane.